So good morning. Today we are going to start a new chapter. We're going to start at the digestive system. Today's lecture is an overview of the processes involved, the movements involved, the blood supply, the nervous supply, and then we're going to take what are called 4 trips through the digestive system. We'll start with the anatomy today, and then we'll do things and add in secretions at a different lecture. So welcome to the digestive system. The digestive system is going to be comprised of 2 groups of organs. The first is the alimentary canal, also known as the GI tract. The GI tract is a continuous muscular tube that runs from the mouth to the anus. It is actually considered to be part of the external environment, which I think is a little wild, but because it opens at both ends to the external environment, it's like we have this tube of environment running through our bodies. Services include digesting food. We'll also talk about things like ingestion, breaking them down both mechanically and chemically. Then we'll move on to absorption. And the organs involved are the mouth, pharynx, esophagus, stomach, small intestine, large intestine, and anus. If an organ is not part of the GI tract, it is part of the accessory digestive organs. We start with the teeth, we move on to the tongue, and then we'll get to the gallbladder and other digestive glands, such as the salivary gland. We'll talk about salivary glands today. And then we'll leave liver and pancreas for a later lecture. So today we stay pretty much esophagus and above, and then we'll move on as the lectures progress. This is a reference figure that you may want to, you can go back to and reference as needed. It shows you the orientation between organs, and then the pathway that food would progress through the organs. I will start with the mouth and the oral cavity and talk about salivary glands, the esophagus. We'll stop our lecture today at the stomach, and then stomach goes into small intestine, large intestine, and then food is excreted, or food waste is excreted. This also shows the accessory organs of pancreas, the liver, the gallbladder. This is the focus of lab next week, will be the digestive system, so that will work out nicely. There are 6 essential activities of the digestive system. The GI tract is essentially a disassembly line. The purpose of the GI tract is to break food apart and extract its nutrients. The first step is ingestion. So we need to eat, we need to ingest food. Then we need to package it in such a way that it can go down that GI tract. So part of the function of the tongue is going to be forming a bolus, which is a small, manageable pouch of food that can then be further digested. So ingestion is going to occur at the oral cavity at the mouth. Then we'll begin with mechanical digestion. We're going to break it apart. We're going to mash it up. We're going to separate it into smaller pieces. Chemical digestion begins in the mouth and will continue through the digestive system. We'll talk about propulsion of food, and that process is called peristalsis. And we're going to spend a little bit of time discussing the difference between peristalsis and segmentation. So the primary purpose of peristalsis is the movement of food. It's like squeezing icing down an icing bag or squeezing toothpaste out of a toothpaste. It's to move those contents forward. Whereas segmentation is if you were to take a bag of icing, hopefully it's still sealed, and then you're squishing it up. If you've got food coloring in there, you're going to try and get it mixed throughout that bag. The segmentation is going to recombine boluses. So it'll break a bolus in half. Some of it will move forward, some of it will move backward, and it will mix with other ones. And then it also turns that food around for better access to enzymes and later absorption of nutrients. We don't want to just get what's on the outside of that bolus. A little bit later, we will talk about catabolism, the chemical digestion of food, and that kind of leads into the following chapter, which is metabolism. Then we have absorption, the removal of nutrients to the blood or the lymph. We'll talk about the structure of the small intestine and how it is optimized for absorption. And then finally, defecation, excretion of indigestible waste. So you need to know each one of these, what this process, the purpose of each process is, but we're going to be discussing each one of these in much greater detail. This is a schematic of the GI tract activities, starting with ingestion. So we ingest food through the oral cavity. We'll start with mechanical digestion with the teeth, and then it follows with churning in the stomach, and then segmentation as it moves down the small intestine. The esophagus is going to, where we'll talk about the swallowing reflex, and then there's peristalsis, the sequential movement of a food bolus down the GI tract. Absorption occurs, it starts as early as the mouth. We'll get some simple sugars there, but then the main location for absorption of nutrients will be in the small intestine. Large intestine is mainly about reabsorbing water and then movement and excretion of waste, but with the final process being defecation. Okay, so this diagram to the right is going to talk about the differences between peristalsis and segmentation. So in peristalsis, adjacent segments alternately contract and relax to propel the food down the GI tract. This esophagus is very good at doing this, so is the small intestine. It's a coordinated movement of contraction and relaxation of the muscles. For segmentation, segmentation has two purposes. It wants to segment and mix up the contents. So it'll split this bolus into two. This bolus could recombine with something coming down through peristalsis. And then it wants to expose as much of this bolus to the cells of the small intestine for absorption. So it wants to break it in half and rotate it. And that will, then it can get better exposure to digestive enzymes as well as cells for absorption. This is also where we talk about the peritoneum, because we are looking, today we're looking at where organs are and how they're arranged in relationship to each other. Organs are going to be mainly in the abdominal cavity, and then we have some that are going to have a lot of movement, and then we have some that need to pretty much stay in place. And it's the peritoneum that accomplishes both functions. So the peritoneum is a double serous membrane that is going to have like so it's a serous membrane system. So all serous membrane systems have a visceral membrane and a parietal membrane. And that parietal membrane is going to line the cavity. The visceral membrane is going to line the organ. And the peritoneum is going to produce a smooth area, like less friction, for when things like the small intestine need to move, because as we have food moving through it, we need it to be able to kind of slide around a little bit in that abdominal cavity. The mesentery is a double layer of peritoneum. So a double membrane next to a double membrane, and then they're fused together to make a pretty strong structure, but it still has some give to it. The mesentery is going to extend from the body wall and support these. It's going to keep like the pancreas in place. It'll keep the liver in place. It'll help keep the jejunum and the ileum in place, because you don't want those too loose. You don't want them to get all twisted and really twisted in on each other. You need some structure. So the mesentery is going to be 2 layers of peritoneum, 2 layers of membranes, so a double membrane that is going to keep organs in place, but also provide routes for blood vessel, lymphatics, and nerves. So the balloon dog is my new kind of favorite example of a peritoneum. A peritoneum is this double membrane system, like a balloon animal. Okay, so you've got 2 layers of membrane, and instead of air, you would have fluid in between those 2 layers. And then it can double up on itself to create structure. That's, I think, the take home of balloon animal, is that if you twist it up and make double membranes, then you can make these relatively strong structures. Okay, so a mesentery is a doubled up double membrane, and it is going to connect organs to the body wall. So it kind of loosely suspends them in the body wall. It also provides a pathway for blood vessels, nerves, and lymphatic ducts. For the peritoneum, there are 3 special types that you'll hear of. One is mesentery. You may also be exposed to something called a peritoneal ligament. Now, I have it in quotes because it's not a ligament at all. It's not made of dense connective tissue with lots of collagen fibers. A peritoneal ligament is a section of peritoneum that has been doubled up, and it suspends a structure like an organ. And then we'll talk about the greater omentum and the lesser omentum later on. But peritoneums are serous membranes, so a pair of membranes with fluid in between. We have a general peritoneum for the abdominal cavity that will have a visceral peritoneum that is around the digestive organ, and then the parietal peritoneum that is going to be around the body cavity. The mesentery is a double layer of peritoneum, so a double membrane, and it will support and suspend organs from the body wall, and then provide this route for blood vessels, lymphatics, and nerves. We're going to take a brief look at the histology of the alimentary canal, and then when we get to individual organs, we will look at how that is either more specialized or slightly changed. In general, all digestive organs have 4 layers called tunics. You do need to know the order. You need to know what is in each one of them. I encourage you to, certainly does not need to be this elaborate, but to kind of create your own drawing of the GI tract, just to keep in mind what the 4 layers are, which are mucosa, submucosa, muscularis externa, and then the serosa. We start, when we describe the layers, we start from the lumen and move, so this would be deep, and then move more superficial towards the outside. Okay, so we'll start with the mucosa. The mucosa lines the lumen, and it has 3 functions: secretion, absorption, and protection. And depending on what organ we're talking about, if we're talking about like the mouth, the esophagus, it's going to be higher on that level of protection. When we get to like the jejunum and the ileum, that's going to be more important for absorption. So the ratios of these functions does change. The mucosa is made up of 3 sublayers. Lining the lumen is the epithelium. It is going to be either stratified squamous epithelium or simple columnar. You'll find stratified squamous in areas that are more involved in protection, like the mouth and the esophagus. You'll find a simple columnar epithelium in cells that are more involved in things like absorption, like the small intestine. There is also secretion, mucus, enzymes, and hormones. We have a whole section on secretions of the digestive system, so we will get there. If you remember, you harken back to 1141, 1147, there should be something in the back of your mind reminding you that epithelial tissues are avascular. They do not have their own blood supply, so they always rely on an underlying connective tissue. In the mucosa, this is called the lamina propria. It is a loose areolar connective tissue through which blood capillaries and lymphatic cells will wind, and nerves wind their way through. Then the third layer, sublayer, is muscularis mucosa. So these are smooth muscle cells embedded in the mucosa. The diagram makes this look like it's a thick layer. It's really not. It's more of a thin scattering of smooth muscle cells, because these aren't involved in movement. Like, they're not going to be involved in peristalsis or segmentation. This muscularis mucosa is involved in aiding secretion and absorption. So it's more a smattering of cells rather than this identifiable layer. Okay, then, so we have the 3 layers of the mucosa: stratified squamous or simple columnar epithelium, the lamina propria to provide a place for blood vessels and nerves to go, and then it should be much thinner than this, the muscularis mucosa, which provides some assist with absorption and secretion. Then we'll move on to the submucosa, which, as you can see from this diagram, is going to be a place where we get arterioles and venules and lymphatic vessels and some nerve fibers as well. And then these are glands. So these glands are going to secrete their contents into the lumen. So the submucosa is connective tissue. It is abundant in elastic fibers. When food is moving through, let's just go with the jejunum, the small intestine, it expands, okay? Well, it needs to come back to shape rather quickly and not stay distended. The elastic tissues of the submucosa are responsible for that rapid going back to into shape. There are also numerous blood and lymphatic vessels, lymphoid follicles, and the submucosal nerve plexus, which we will talk about as part of the enteric nervous system. Moving on out, we have the muscularis externa. It has 2 layers. Usually, and this is something that can kind of, like when we get to the stomach, we'll talk a little bit more about how its layers are arranged. But in general, the inner layer are going to be circular, so surrounding the lumen, with the outer fibers are longitudinal. The muscularis externa is responsible for the movements of peristalsis and segmentation. So these are true, a thick muscular layers. Sometimes it's skeletal muscle, sometimes it's smooth muscle, depending on the organ. The serosa for most organs is going to be the visceral peritoneum. So we talked about those serous membranes of the peritoneum. In certain organs, it is replaced by the something called the adventitia. For blood supply, we are interested in two circulations. The first circulation is called the splanchnic circulation, and that is going to provide oxygen to the digestive system. So there are arteries that branch off of the abdominal aorta. We have that celiac trunk, if you remember, kind of looks like this, of which the hepatic, splenic, and left gastric arteries branch off of. And then we also have the inferior and superior mesenteric arteries branching off as well. The second circulation that we are going to be discussing is the hepatic portal circulation. So the hepatic portal circulation is low in oxygen because it goes from a capillary bed to another capillary bed. So this is going to be low in oxygen, but high in nutrients. And we will discuss the hepatic portal circulation when we get to the liver. Okay. So two circulations. Splanchnic arteries deliver oxygen. Hepatic portal vessels are going to deliver nutrient-rich blood from the digestive organs, mainly from the small intestine, and then to the liver for processing. Moving on to the nervous system. The GI tract has its own nervous system. This is called the enteric nervous system. The enteric nervous system has about 100 million neurons. So that's more than the spinal cord, but much less than the 86 billion neurons of your brain. Okay, so 100 million is good, 86 billion is a lot. There are two nerve plexuses. Okay, so the nervous system of the digestive system is called the enteric nervous system. There are two nerve plexuses in the enteric nervous system. The first one is the submucosal nerve plexus, and it is going to regulate glands and smooth muscle in the mucosa. And then the second one is the myenteric nerve plexus. And this is going to usually it's in between the circular and longitudinal layers. So it's in that muscularis externa tunic. Okay, so we have a nerve plexus in the submucosa and one in that muscularis layer, the external one, the big one. These two nerve plexuses are going to promote very different functions. The submucosal, so we're going to have submucosal and myenteric for the nerve plexuses. These are going to participate in what are called short reflex pathways or long reflex pathways. For short reflexes, these are going to occur entirely within the GI wall. So the short reflexes are going to be peristalsis and segmentation. And the nerves that are involved in short reflexes are going to be those in the myenteric plexus. There are also, we have that submucosal plexus that is going to participate in long reflexes. And these are going to be for the autonomic nervous system, we have control by both the parasympathetic and the sympathetic divisions of the autonomic nervous system. So depending on what we're doing, okay, so we've had Thanksgiving dinner, we're now sitting in our recliner and getting ready to watch some TV. The parasympathetic division of the autonomic nervous system is going to send signals to this submucosal plexus to promote digestion. So it is going to target smooth muscles and glands and increase contractile or secretory activity. If we decide that we need to go for a run, maybe hopefully before Thanksgiving dinner, then these sympathetic fibers are going to then inhibit smooth muscle glands and inhibit contractile and secretory activity. So we also have external stimuli that can send information to the central nervous system. This is why we start to salivate before we even start to eat. But the take-home messages of the enteric nervous system are that we have these two nerve plexuses, submucosal and myenteric. Submucosal is going to interact with the parasympathetic and sympathetic divisions of the autonomic nervous system with long reflexes to affect the activity of smooth muscle and glands. Then we have the myenteric plexus that is going to be involved in short reflexes in order to control peristalsis and segmentation. For regulating digestive activity, there are three key concepts. The first one is that we need to detect the presence of stimuli. So we need to detect that food is present. And this is accomplished by mechanoreceptors, which respond to stretch. And this is important in places like the esophagus. It'll be important even in places like the stomach. The stomach actually, so it has things called rugae, and can be quite small, okay, because the rugae allow it to fold extensively. But then as food is deposited into the stomach, it is going to increase its size. When it increases its size, then stretch receptors are going to start secreting more pepsin, more digestive enzymes and hydrochloric acid. So to start digestion. So as the stomach stretches, those mechanoreceptors send visceral afferent stimuli. Don't worry about that too much, but visceral signals that are going to stimulate secretion by glands. Other cells are more susceptible, they're more sensitive to changes in either osmolarity, so salt concentration usually, and or pH. So the stomach contents are very acidic, and we will need to neutralize that using digestive enzymes. So we will have osmoreceptors as well to monitor that process, and chemoreceptors. So mechano and chemoreceptors detect stimuli all along the GI tract. So that can be due to stretch, it could be due to changes in pH, presence of substrate or end products of digestion. Okay, so once we detect stimuli using receptors, then we need to respond to the stimuli. Certain receptors, like in the esophagus, are going to respond by propelling that bolus through the GI tract. Other ones are going to stimulate smooth muscle to mix, so segmentation, or we have churning. So there are going to be physical processes. Reflexes, like we just talked about, can activate or inhibit digestive glands. We may need more or less pepsin, we may need more or less bicarbonate. These are going to be in response to the stimuli that they're receiving. Okay, so we detect stimuli, we respond to stimuli, and then we need to control the actions. And those controls can either be through the nervous system, through that enteric nervous system, using short reflexes, which these involve primarily that myenteric nerve plexus, or extrinsic controls, which are going to involve the submucosal nerve plexus in coordination with the sympathetic and primarily parasympathetic divisions of the autonomic nervous system. There are also hormonal controls. Again, if you harken back a semester, you might remember ghrelin or gastrin or cholecystokinin. We'll be talking more about those when we get to metabolism, that are going to target cells to aid in digestive processes. Okay, so now we've kind of given that broad introduction. So that was called part one. It's the broad introduction to the orientation of digestive organs to each other, the four tunics that are throughout all of the digestive organs, which with modifications, which we'll discuss later. We've talked about the two circulations, the splanchnic circulation, which is oxygen rich, the hepatic portal circulation, which is nutrient rich. We've talked about the enteric nervous system, how it has two plexuses, submucosal myenteric. Myenteric works through short reflex pathways. Submucosal will interact with the autonomic nervous system on longer reflex pathways. And so now we get into what are called parts 2 and 3 and maybe 4. I can't remember how many parts the chapter is broken up into. I know it's at least 3. Okay. So for the next 2 lectures, we are going to go through these trips through the digestive system. So there are going to be 4 times where we are starting at the mouth and going down to the anus. And then we come back and we'll talk about the movement from top to bottom, and then secretions from top to bottom, and then digestion and absorption of nutrients as well. Okay, so there's a lot of like circular redundancy in this chapter. Okay, but we feel like we should start with the anatomy and we'll go from there. Okay, so trip 1, the anatomy of the GI system. And we will not get through even the anatomy today. But we're going to start at the top. We're going to start with the oral cavity, also known as the buccal cavity. It is bounded by the lips, cheeks, palate, and tongue. It is continuous or contiguous with the oropharynx. So the pharynx has 3 sections. It has the nasopharynx, the oropharynx, and the laryngopharynx. The nasopharynx is not considered part of the digestive system because food should not be going up there. For the soft palate, its purpose is to prevent that from happening. The mouth has largely a protective function. So the mouth is lined with stratified squamous epithelium, and parts of its boundaries, like the gums, the hard palate, are slightly keratinized. Not quite like your keratinized skin, but they do have some keratinized to toughen them. So we have the oral cavity. We're going to talk about the hard palate, the soft palate, the tongue, and then we'll circle back to some salivary glands. The hard palate is hard because it is made up of bones. It is made up of the palatine bones, as well as the palatine processes of the right and left maxillary bones. So the hard palate is made up of bone, and it is lined with a slightly keratinized epithelium. This slightly keratinized epithelium is going to give it corrugations, so little ridges that the tongue, when it pushes food up against, it gives it friction to work against. So we don't want food sliding either on the surface of the tongue or the surface of the palate. So the palate is going to be slightly ridged or corrugated, and the tongue is going to have things called papillae to give it a friction surface to work with. The soft palate, I actually still like this picture better. The soft palate is made up of muscle and epithelium, and it ends in a tip called the uvula. And its purpose is that when the swallowing reflex is initiated, it will come up and close off the nasopharynx. So that the pathway of a food bolus is down through the oropharynx, laryngopharynx, and then to the esophagus. One of the things that the book suggests that you try is breathing through your nose and swallowing, and you can't do it because if you're swallowing, this pathway is going to be blocked off as it should be. Okay, so epithelium and muscle, it is going to close off the nasopharynx during swallowing. Okay, so on to the tongue. The tongue is going to occupy the floor of the mouth. It functions to grip and reposition food during chewing, mixes the food with saliva and forms the bolus. So the bolus is this smooth ball of food that will get propelled through the GI tract. It initiates swallowing, and then it is involved in speech and taste. There are two sets of muscles, skeletal muscles, for the tongue. Intrinsic skeletal muscles are not attached to bone, and they can change the shape of the tongue. So, some people can like put their tongue like a little taco, and some people can't. Those are intrinsic muscles that are controlling that motion. Speech, especially the formation of consonants, is involved with the tongue, and then swallowing. Extrinsic skeletal muscles for the tongue are going to allow the tongue to protrude. So if you want to stick out your tongue, if you want to retract or pull it back in, or if you want to wiggle it from side to side, those movements are controlled by the extrinsic muscles. So intrinsic, speech, swallowing, and then extrinsic, actually moving the tongue around. Okay. I always get this picture of rock bands in my head when we're going through that slide. Okay, so moving on to taste buds, and these are 3 of the 4 types of papillae. So papillae are little protrusions of tissue on the surface of the tongue. There are 4 sets of papillae. The filiform are small but they are numerous. Okay, so they are they're going to be everywhere on the tongue. They're these little things all over the tongue. And their purpose is for friction. The second set of papillary are the fungiform, because they thought that these things looked like little mushrooms, mushroom heads. Oops, I got a little big. Valate, I don't know what how that one got its, I don't know what that's derived from. And foliate looks like a leaf. Okay. So fungiform, so leaves and mushrooms and whatever valate comes from are going to house the taste buds. So we have filiform for friction, fungiform, valate, foliate for taste buds. Moving on to saliva. We're going to talk about the composition of saliva when we get to secretions. But the functions of saliva are to cleanse the mouth, dissolve food for chemicals for taste, moisten the food to allow it to compact into a bolus, and then also is going to begin the breakdown of starch because the enzyme amylase is secreted in saliva. But again, we'll go over the composition when we get to secretions. Most saliva is produced by the three major extrinsic salivary glands. So these are very large salivary glands located outside of the oral cavity that then dump their contents into the oral cavity. There are also some minor salivary glands called buccal glands that are scattered throughout the oral cavity. The three main, and this will be part of your digestive lab, is locating these three sets of salivary glands. The largest is on the side of the cheek, okay, here. And these are going to be the parotids. They're associated with the masseter chewing muscle. So the parotid salivary glands are very large, and then they will dump their secretions in through ducts. Then we get the next largest would be the submandibular. So it's located here, kind of underneath the grooves of the mandible. And then we have the sublingual underneath the tongue, which actually has the most glands. It's got a lot of glands that kind of poke up underneath the tongue. All three of these categories of salivary glands, groups of salivary glands, are made up of serous cells and mucus cells. The parotid and submandibular are mainly serous cells, water secretion with enzymes, and a little bit of mucin. Mucin is a glycoprotein that gets sticky when wet. The sublingual glands will produce mainly mucus, less enzymes and more mucus. Now moving on to teeth. This is always a fun topic. We have two sets of teeth. We have deciduous teeth, also known as our primary teeth, and babies tend to start with their central incisors. Teeth are named by their shape and their function and their location. We're not really going to go into tooth structure. We could spend a whole class on teeth, and we're just not going to do that. I know it's a little sad because some of you are like dental assisting programs, but hopefully I'm sure you'll get all of that in your classes. So the central incisors are usually the first ones to come in, usually on the bottom. These start to come in at around 6 months. I know some babies get them much earlier and some get them much later. The last primary tooth usually comes in during the second year of existence. Okay, so there are 20 total. Then there are 32 permanent teeth. So the roots on the primary teeth, they get reabsorbed, and that causes the primary tooth to fall out. And then the tooth fairy comes and leaves a nice little present underneath. And the permanent tooth grows up underneath it. Sometimes they grow up behind it, and then these have to get pulled. But ideally, these get reabsorbed. They fall out, and then they get replaced. The first replacement usually occurs around age 7, maybe a little earlier, but kinder, first grade, somewhere in there. The last set are going to be the wisdom teeth. These don't erupt until 17 to 25 years of age. And in theory, they were probably meant to before modern dentistry. Most individuals would have lost at least one on each side of the other molars due to decay. So these were kind of more replacement teeth, which is probably why most of us don't have room for a third set of molars. So many people will have these removed. Okay, so 32 total permanent teeth. Moving on to the pharynx, then we'll get to the esophagus. How are we doing on time? Oh, we're doing good on time. Okay, so the pharynx, as I discussed, has 3 sections. The nasopharynx, which is not a part of the digestive system because food should not go up through your nose. The oropharynx, which is where food comes through the oral cavity, starts to make its way down from the oropharynx to the laryngopharynx, where it gets shuttled because of the epiglottis down to the esophagus. So under normal circumstances, when you're not eating, you're just talking and breathing, the epiglottis is going to be open. This is going to, it kind of closes flat. And then air is allowed to come down into the trachea. But when we swallow, the glottis moves up, closing off the trachea, and food is routed down the esophagus. As oro and laryngopharynx allow passage of food and fluid to the esophagus and air to the trachea. It is made up of stratified squamous epithelium, mucus glands, and skeletal muscle. The laryngopharynx is going to merge with the esophagus, which is a very strong muscular tube that leads to the stomach. Travels through the mediastinum, pierces the diaphragm, and joins the stomach at the cardiac orifice. So one of the things that we will discuss are the sections of the stomach. So we'll talk about the cardia, the fundus, the body, the pylorus, the pyloric sphincter. That's all coming. The esophagus is lined with stratified squamous epithelium for protection. And when empty, it is going to fold in on itself. And then it flattens out when food is moving through it. There are glands that secrete mucus to assist in the bolus movement. And that movement is called peristalsis. The peristalsis is the movement of the food bolus down the digestive system. The muscularis externa layer is going to change. So this is kind of interesting, I think, and that the esophagus is a transition zone. The muscularis layer is going to change from skeletal muscle up by the pharynx. And then as it gets closer to the stomach, it transitions to smooth muscle. So the esophagus is skeletal muscle and then smooth muscle. I think that's really interesting. It is going to join the, so the esophagus comes down, Then there is a layer of smooth muscle that creates the lower esophageal sphincter, also sometimes called the gastroesophageal sphincter. I kind of like lower. I think it's what you'll see more often. The lower esophageal sphincter, its purpose is to prevent heartburn, essentially. So our stomach is going to have digestive juices, stomach acid, be about a pH of 2. In order to prevent that acidic fluid from going up and burning essentially our esophagus, that lower esophageal sphincter stays closed to keep the digestive juices in the stomach. If this gets loose or gets disrupted by, like, there's too much food or there's movement or something, then that's what heartburn is, where the digestive juices escape up through that lower esophageal sphincter and burn the esophagus. We'll just touch on the stomach because that's where we're going to pick up at the next lecture. So food is going to come down through that lower esophageal sphincter, and then it is going to enter the stomach. The stomach is comprised of the cardia. So the cardia is this section where it joins with the esophagus. There is also the fundus. The fundus is the part that is above this sphincter. Okay, so this part up here. Then we have the body here, and then the pylorus is going to be down in here. We have the pyloric sphincter, which is going to regulate the movement of chyme, so movement of like digested food into the duodenum. And the purpose of this pyloric sphincter is to not overwhelm the duodenum with acidic contents, because we'll get base excreted. We'll talk about that later, to neutralize the contents, but we can only secrete a small amount at a time so that we don't also burn the duodenum. Okay, so again, all of that kind of gets covered at a later, sorry about that, a later date. I think I need to learn to bring a water bottle with me. Okay, that is where we're going to stop for today. So the next lecture, we pick up at the stomach. We're going to start at the stomach and we'll finish anatomy, and then we'll move on from there.